record. Cool. All right. Cool. So I have the great pleasure to introduce Zack. Zack's a former coworker and an excellent engineer, and also one of the developers of the eVOLVER continuous culture system that comes out of Khalil's lab at Boston University. So Zack currently is working in industry, but is going to talk to us today about how that open source tool has worked, and I'll let him tell you about it. So Zack, take it away.
Thanks, Tim. Yeah, can everybody see my screen?
Yep.
Let me hide the meeting controls. All right, is this working? You guys seeing the right thing here? This always messes up. Okay, great. Yeah, so thanks, Tim, for the introduction. So yeah, my name is Zack. I did my PhD at Boston University in Khalil's lab, primarily working on the eVOLVER continuous culture platform, which I'll talk a bit about today. And yeah, let's just jump into it because I think I got a lot of stuff here. I kind of don't want to go along here. So just as a little bit of background, growing microbes in liquid culture is, pretty core part of the biotechnology sort of way things work in both academia and industry, right? So if you're thinking, there's been like a huge boom in the way we can modify genomes and create strains that can do all kinds of really cool things. The way you characterize that is you primarily grow them in liquid culture, right? And so there's two main ways that this is kind of done. You have like what you see on the left, which is a bioreactor, which is a very complex piece of equipment, very expensive, but has very, very tight control over all the parameters. So you know exactly what's going on in terms of like temperature, DO, pH. You can control kind of anything you want within this system, very fine-tuned control. On the other end of the spectrum, you have something very high throughput, right, which is like batch culture in plates or wells, where you can test out lots of different conditions all at once, but you might not necessarily get as rich data from that, like in terms of real-time continuous data, and you can't interface with that system on the fly as easily. I think it's getting a lot better with a lot of the work people are doing in the automation community, but traditionally, this is kind of how things are kind of done. So if you want to understand sort of the phenotypes that arrive from different types of environments that are placed on these cell cultures, you need to have the ability to control lots of things all at once and dynamically, right? And so like let's just say in synthetic biology, I've made some sort of circuit and I want to know how it's going to behave in different environments, right? To do this sort of experiment, I'd like to be able to grow it in lots of different ways. So I need to be able to control these types of things to sort of map out what that fitness landscape might look like. And so eVOLVER is a tool that was sort of built and designed to sort of meet this need. It's an open source automated platform that lets you scale and automate laboratory cell culture to sort of try to fit this sort of in between of having control and continuous data, but also not being prohibitively expensive and complicated and hard to use, but also giving you sort of higher throughput. Oh, this is a video, I forgot. Oh, sorry. That's going to disappear and then it won't show the video. That's fine. But on the bottom, you can see kind of a picture of what this looks like at the Khalil Lab when it's scaled out fully. I think it's grown since then, since this picture was taken. So this is like a whole bench that's set up. Each one of those systems has 16 vessels on it. They're all being controlled over the network on one computer. So you can kind of, and each one is individually programmed too. So you can have all kinds of different conditions. You can run replicates. Yeah, kind of do whatever you kind of want at a higher throughput, at a higher scale than what's possible with like, say, a bioreactor. But you have more control than like a batch culture that you just kind of throw in an incubator. So how this works is the sensor, you have different sensors and actuators and electrical components, which are attached to this device, which is called the smart sleeve. The vessel is around 20 to 40 milliliters. There's work currently ongoing in the lab to go smaller and bigger. So if you need like larger library sizes or larger culture for whatever reason, there's a platform that's being developed for that. There's also a platform that's going smaller so that you can fit even more of these things on a single platform and do even more control. But the gist of it is you have this small culture, you've got diodes and PID controlled heaters, just thermistors and different ways to agitate the culture. And there's even development going on, which I'm not going to talk about too much, but there's development and controlling the gas concentrations in the headspace for doing like anaerobic culture. If you're interested in like gut microbiome, that's important. So you're doing all these sorts of things, you can control them over time. And it's sort of trying to strike that balance between controllability and throughput. Also not kind of pictured as complexity, cost, these kinds of things, which also are extremely important in academic environments. Like you can't afford expensive, hard to maintain equipment as easily, but a small system like this that fits on the bench is pretty easy to set up and use. A key thing is that this is designed to be customizable and scalable. So this video on the bottom is just, I think, a lab tech just putting one of these things together. All the parts are easy to get from. They're just like off-the-shelf parts for the most part. There's also 3D printed parts, so it's kind of easy to get all the things you need to build these yourself if you wanted to. And if things break, spills happen, things break, not a big deal to repair them. And they're also designed to be customizable. So like you can build these to kind of whatever specifications you want if you have a different kind of need. It's not too hard to do. So how the platform sort of works is we have lots of arrays of these things, and there's a coupling at different layers. So we have at the one end of the spectrum, you have the sensors and the actuators at the smart sleeve, which are controlled by Arduino microcontrollers, which is customizable. You can do kind of whatever you want with that. There's a set of parameters that are provided out of the box, but people are making all kinds of different stuff to do different kinds of things. Those serially stream data to a Raspberry Pi. That's all Python software that's controlling the actual communication between telling the actuators and the sensors what to do, collecting data that goes to a Raspberry Pi. There's Python that's then streaming that over a network to a lab computer that actually does your control. So if, like, let's say you want to have a temperature profile, I want to do a sinusoidal temperature wave, you can program this in Python pretty simply. The code is very accessible. That was one of the main kind of things that we tried to do with the design of the software was it needs to be usable by biologists. We didn't want it to be like you need to have like intense software engineering, electrical engineering background to be able to do this. Like the point is we want to get this hardware into the hands of the people who have these creative biological like experiments and ideas who are otherwise limited by technical capability. Getting them a tool that's easy to use was the idea. So we've made like everything is in Python for that reason, to try to make it simple. There's libraries that are provided so that you don't have to write any sort of low-level code. You can just say fluidics, send pump for 5 seconds to this vial, and it'll do it. Yeah, here's just another picture of of kind of what it looks like. You can kind of see the decoupling between these things. Another kind of tenet that was in the design is that things are standardized footprints. So if you wanted to do some sort of new hardware module, let's say you want to make a fluorescence detection unit or something, you can build your own kind of custom hardware, and it slots into the motherboard. You can do different, there's multiplexers, there's ADCs, there's PWM modules. I've made serial communication modules. So if you wanted to have some sort of thing that I just want to talk to 16 vials, you can pop in a thing onto the motherboard to do that. And then that would communicate with the rest of the system. Okay, so I'm going to go through a couple of examples of how we've used this in the past. So one of the most simplest ones is, let's say you want to generate a fitness landscape, which is a pretty common thing in synthetic biology. I just want to know, I want to compare this strain I made to a wild type in a range of different conditions, right? So your inputs of this would be, I have my cells and I have my selections and stresses that I want to apply to these cells. With eVOLVER, you can program some sort of algorithm to modulate across all your vessels, that landscape, just from low stress to high stress in multi-dimensions. And then your outputs are, the system is streaming data. You can collect cells and do any sort of offline analysis that you want as well. And so this is just a little more detailed example of this specific case. So in this case, we did two things, which was heat stress. So we just varied the temperature from 30 degrees to, I want to say 40, 45, I can't remember exactly. It'll be in the data, but we varied the temperature, and then we also varied the salt concentration to see how these strains that we are comparing against were able to handle these two stresses in combination. And so you can see in the algorithm there is that we have pretty much we're changing the temperature in the stir, and we're also controlling the optical density, so the turbidity of the cells, by keeping it between a defined window. So all the cells are seeing kind of the same sort of environment in that regard. We're just modulating the temperature and osmolarity. And you can kind of see representative data of what it looks like. One thing I'll also point out, the way you do this is we have two different, to compare how it's doing. We have a reference strain, which we've labeled with the GFP fluorescent reporter, and then you have the variant. And so if you take a sample and you look at, if you started out with an equal population, and then after a certain amount of time, that population, you see more GFP than you did than non-GFP, you know that your cell is less fit, right? And so the cool thing is, like, doing this in eVOLVER is you can program this to happen through the Python software. And this is kind of an example of what the data looks like. This is from one eVOLVER. We did three cell line or three sets of experiments. So a wild type versus wild type, a temperature sensitive strain and a salt sensitive strain, and 16 replicates across this whole thing. So 48 files total. This is just what the data from one eVOLVER might look like. And all this data is being streamed into dashboards. These dashboards are kind of evolving over time. So I made this data just to look pretty and rainbow for a paper, but the system does come with graphing software out of the box. And there's currently work being done to make that a little bit better. People are also making their own stuff. All this stuff is open source. So people are making dashboards to view. Like, there's a lab that's measuring how fast their media is running out over time so they can track their media consumption. There's labs doing things that they've integrated single cell flow cytometer data into their own eVOLVER. So, people are making dashboards for all kinds of different things. This is a really simple example. I'm just looking at turbidity over time. And from this, you can extrapolate growth rates, how many generations the cells have gone through as a function of time and as under these different conditions of stress. So finally, this is the final figure, for this contrived sort of simple example. I have these three cell lines. How do they kind of look under these different stress conditions? And you can see we calculate this fitness metric, which is basically the slope of how fast are you growing compared to the wild type over time. And you can make at the bottom right there, you can see what it looks like how these cell lines behaved under these different stresses. So it's a really simple example to showing kind of a simple kind of thing you might want to do with eVOLVER. This is a more sort of relevant, interesting example. So this is a paper that came out in 2023 last year from someone from the Khalil Lab. There's a really cool paper about characterizing synthetic transcriptional regulation and doing signal processing in cells. And so one of the things that they tested with eVOLVER was just genetic stability, like showing how these synthetic transcriptional regulators that they made can maintain high fitness while also having high activation of a circuit, which is a problem in a lot of synthetic biology problems. I make this circuit that can activate some gene pathway, and it does a really great job if I just do one sample point. Like, I make my thing, I induce it, I get good data, that's great, one-time measurement, not stable over time, which might be important in a certain context like biomanufacturing, where I want to have my circuit be stable for a long period of time, from days possibly, right? So this system that they designed allows them to have this maintain high fitness while also having high activation, not losing the circuit over time. And this was just tested in eVOLVER. They just grew these over time. You can see for 120 hours. Comparing at the top is just one system where it's a high binding affinity synthetic transcription factor. You can see the fitness drops very quickly. That's the cell line mutating, losing the circuit. And you can see the synthetic transcription factor expression in the second column also dropping down in the circuit, on the right also decreasing. If you have a low affinity binder, the fitness stays high, but your activation is low. And so this synthetic system we made maintains high fitness, but also has high activation, which is cool. So this is just an example of characterizing your circuit over time. Okay, so I'm going to switch gears now to something that my work was more. This is what I kind of focused on during my PhD, which was directed evolution. So this is another use case for growing cells, is you can study evolution, right? You can study and you can actually use evolution as a tool to design novel functionality in proteins. And so if you think about a typical protein of around 300 amino acids, if you were to think about all possible permutations of this space, that problem is incredibly crazy, right? There's so many different types of mutants. Doing this in a rational design sort of way is not really possible. And I love this example. This is an example that Francis Arnold, who's one of the pioneers in the field, sort of pointed out, is this example of the Library of Babel. It's a short story that's just, the idea basically is that there's a library that contains every possible sequence of words or characters that can be, right? And so if you pulled any one book out, it's most likely going to be nonsense. But in this library also exists all the works of Shakespeare, all these brilliant things, because it has every possible combination. So it's a good way to think about rational design of proteins, is that, this is not an easy kind of problem to do without some sort of tool. And so evolution is a tool we can use to kind of solve this problem. You can think of it as a fitness optimization process where if you think of sequence on your X and Y axis and fitness at the Z, what you can do is sample around a certain point, find something that has a higher fitness, use that as your starting position for the next sort of round of like sampling around that space, find the next one, you just keep kind of moving until you find a solution to your problem of whatever new kind of behavior you want. And the problem is that some of these landscapes can be really, really rugged, right? So it's not this, like on the right, you see this like nice, beautiful, smooth landscape. That's a pretty easy thing to do, right? I can sample around a space, test my fitness, see if it's better, and go from there. It's much harder when it's rugged, where like small changes in your sequence like can cause you to like drastically increase or decrease fitness, right? Oh, I forgot to have animation, sorry. There you go. So a way to do this, this is a, here, I'll go to the next slide actually, because I think it's, we'll skip through this. I don't want to spend too much time. There's a lot of cool things you can do with this tool, basically, is kind of the point of this slide. But this method has been evolving over time. So when this was first made, this was pioneered by Francis Arnold and others, and it recently won a Nobel Prize. The basic way you do this is that you take a gene of interest, you do some sort of way to introduce mutations randomly throughout the protein. PCR mutagenesis is one way. You clone those variants into a cell line, and then you screen them. So you just test like which one of these performs the best in terms of like my new sort of functionality that I'm sort of trying to push to. And you do this over and over through these discrete screening steps. And this process takes, many weeks to do, as you know, if you're doing this process of cloning, transforming, screening, cloning, transforming, screening, that's a slow process. Like it takes a lot of time, a lot of manual labor. So advances to this have been moving towards these systems that use continuous methods of doing this. So instead of these discrete steps, you have a constant way of introducing mutations into a protein and a constant way of selecting instead of screening for variants that have increased activity. And so there's two prominent examples here, PACE and OrthoRep. I'm going to talk mostly about PACE, but we'll get to that in a minute. So why this is better, why this is making things better. One is that you're increasing the scale, right? You're able to sample just more spaces, more things in this fitness landscapes, right? Because you're continuously mutagenizing your population. You're constantly sampling around this different space. You find more things faster. You're also able to find increased depth. And that means, what I mean there is in terms of evolutionary distance, right? So I can, because I'm continuously screening and doing this, nonstop as opposed to discrete steps, I'm able to move further in evolutionary space by using a continuous method. And lastly, and most importantly, is speed, right? That's the main reason why this is better. Instead of something taking weeks to do, I can do this in a couple of days, which is ultimately the goal. Okay, so PACE, you don't need to know this too much. This is just like the platform that we used. The gist of how it works is that you have a bacteriophage that you removed a gene that is critical for it to replicate, and you put it inside of E. coli, and you put the transcription of that gene under some activity that you care about. And so what happens in PACE is that you have this vessel called the lagoon where your phage resides. You're constantly flowing in your cells, E. coli cells. The phage will infect, and they will also mutagenize. You have a mutagenesis plasmid in there, so they'll introduce mutations, they'll infect. And then there's a couple of things that can happen. One, you can either have an inactive protein of interest, right? So the mutations that the phage have accrued are not beneficial. They will not be able to replicate as efficiently as ones that do, and they will ultimately just be flown out through waste. So you're constantly flowing cells into this vessel in and out. Those will just get washed away. If, however, the phage gets some mutation that's beneficial, they will be able to replicate. They will replicate faster and more than ones that do not, and they will continue in the cycle of infecting these cells coming in. And so through this method, you're able to generate mutations and also screen continuously for things that are higher activity. Okay, so here's the meat of why this is interesting from an automation perspective. This method is super, super tedious and challenging the way it's traditionally done, to such a degree that really only one lab does this, and that's David Liu's lab out of the Broad. No one, and some of his his progeny, if you want to think of it that way, have also do this method. But not many people really pick this up because it's kind of hard and annoying to do. You need classically, traditionally, what they do is you have a dedicated room for this, which is a heated warm room, which is really gross to work in. You need these expensive pumps and syringe pumps, and just doing continuous culture in general is kind of tricky. Like it's not, it's getting better, but like it's not an easy thing to do just by itself. And then also adding on top of it this complicated selection system, it's just a really hard problem. And so what we did was made this platform called ePace. So this is bringing together eVOLVER, which is the platform for doing continuous culture, and Pace. We made a system that can do these things together on the bench, and at higher scale, higher throughput. Something I didn't mention is, evolution is a stochastic process. If you have this one room that you have to dedicate to this, and you have these pumps that you have to manually control, you really can only do 1 or 2 replicates at a time, which is a problem if you're trying to well, I'll demonstrate why this is a problem in a second, but you want to be able to do more of these things at the same time. Cool. So I'm going to get into like what we use this tool for. So CRISPR-Cas, precision genome editing, is, I'm sure many of you have heard of this. The way it basically works is you have a Cas9 molecule with a guide RNA. You can direct an effector domain like a base editor to a region of the genome, make an edit. Very useful for potential therapeutics, also just generally useful for synthetic biology, for making different cell strains. You can do all kinds of really cool stuff with this tool. An example of like just a more therapeutically relevant example is this is a case where base editing rescued this progeria. It was able to cure progeria in mice. So progeria is a syndrome that it basically has you rapidly age. If you have this condition, it's pretty rare, but it's also in humans. You rapidly age and you also end up with heart conditions. And so you can see in the videos, untreated mice after 7.5 months, not looking so good. This base editing technology that they made was able to basically cure these mice so that they have much better lives. So there's a lot of potential for this base editing technology to be used in therapeutics for humans. But the problem with these things is that you need to have very, very precise Cas9 molecules to make it as a therapeutic. You can't have any off targets. Like the way you do these edits is very, very important. And if you don't have throughput, making these molecules is very hard, especially if you're using a system like Pace to do it. So the way that this works is you have your Cas9 that you're evolving. You try to push it towards a different region of the genome that you want to be able to edit. And so the recognition of this PAM sequence is the most important thing. I'm blasting through this. I'm sorry. If you have more questions, we can talk afterwards. I'm trying to go kind of quickly here. But the key limitation of Cas9 technology is this PAM sequence. So it's a sequence that the molecule will recognize in the genome that must be there in order to make an edit. So if there's not a PAM sequence next to like a spot in the genome that has a mutation that you need to change, right? If you're trying to edit a spot in the genome and there's no PAM sequence, you can't target that spot, which is a problem, right? And so a paper, this came out in 2018, is where they tried to expand that capability of Cas9 by evolving on a pooled library of like lots of different PAMs. So like every possible PAM for SB-Cas9, right? Just specific Cas9 molecule. What ended up happening with that is that instead of getting a molecule that could bind pretty much anything that you wanted, you ended up with a molecule that was sort of high activity to moderate activity on a subset of those PAMs, where there was little to no activity on a lot of the other ones. And lots of research came out after the fact too that showed, this molecule had really bad off-target problems too, right? So if you wanted to target a specific region of the genome, you might end up making edits all over the place at places you didn't actually want to happen, because it has this limited, it doesn't have this very stringent PAM restriction anymore. It's able to just kind of bind kind of wherever. So that's a problem, right? And so not having throughput is kind of the reason that they had to do this strategy to begin with, where they pooled all these PAMs together and evolved on a pool, as opposed to the strategy that I'm going to talk about, where instead if you have throughput, you could evolve on specific PAMs. So you can try to push it towards, let's say I have a specific region of the genome I want to target. I can evolve to target that specific spot instead of trying to make a general purpose molecule. And so this is a example. The the method that was developed, this is the synthetic biology side, was sequence agnostic Cas9 PACE. So this selection requires PAM recognition and base editing activity to happen. So we're evolving in a more specific way. We're not just evolving for PAM recognition, we're evolving for activity as well. This is like super fast, but I'm trying to go quickly here. So basically what we ended up doing is evolving on a specific PAM over many different runs on using ePace, right? So ended up over the course of about 2 months, I say, we ran about 5 to 6. There were a few other PACE experiments that aren't pictured here, but about 5 to 6 experiments running in duplicates on all these PAMs with eVOLVER, right? And so in the middle, you can kind of see we started with a wild type where you can see it has activity at one specific region, right? This NCC region. And over the course of our experiments, we were able to evolve 3 molecules. So E-enemy-2C, which is the third from the top, which has broadened activity on any C-containing PAM. So it's basically 4NC is what it targets. And then an E-enemy-2T, which can target any T region of the genome. And you can see on the right that these have increased activity in comparison to wild type and other molecules. We also then subsequently took these variants and showed that they're efficient in human cells. You're able to target these ACC, CCC, any C-containing region of the genome much more efficiently than was possible before. And we also were able to show that you can target disease-relevant mutation sites that were previously not targetable with existing Cas9 technology. So this specific spot is the RBM20 locus, which is a mutation that's associated with familial dilated cardiomyopathy. So we were sure that these molecules were able to make mutations in this gene that potentially has therapeutic importance, right? So this is, and we're comparing to existing there's another Cas9 that's like nearly what do they call it? It's the way they framed it was that it can target almost any PAM, like it doesn't have a restriction, showing that this has better activity. We also have data showing us lower bystander, lower off-targeting problems than these other variants. So, which is cool. The other cool thing about this is that just to kind of demonstrate why this would be a hard problem to do with rational design, the mutations that were found were actually all throughout the protein, not just at this region that interacts with the DNA. It required mutations like throughout the protein, right? So this is just highlighting again how evolution is a cool tool because you don't necessarily have to understand, everything about this protein, about like how conformational changes might happen. You can just sort of let evolution solve the problem for you to find out what's important for this protein for the activity that you care about. So this is sort of, that was the paper. This is sort of work that's gone on since then, is trying to make this technology more accessible. And so this is something I made. The graph on the bottom right was actually the last experiment I ran for my PhD. But what we did was we took, the existing eVOLVER platform and we sort of miniaturized it. So I made a new board that's, as you can see on the bottom left, very, very small. It has enough, it has the capability of supporting 6 pumps and then 2 vessels. So you can do like a single PACE experiment with this module. And why this is cool is because, while yes, having the extra throughput is really important for a lot of evolution campaigns, there's also a lot of times when you just need to be able to run a PACE experiment, right? And like not having to have a dedicated room and like you have to schedule time on this equipment, having these little benchtop systems that are really cheap to make and like throw anywhere is really powerful for the labs that already do PACE and also other labs who are picking up PACE. So this has been, this work's been continued by the Khalil Lab. I think they're giving these out now to the different labs like at Tufts and Scripps and other places too. Like it's becoming pretty popular. It's like a simple way to sort of get PACE off the ground without needing all this expensive equipment. And so the bottom right is just an experiment showing, validating that, yeah, you can do PACE. We were able to evolve a protein-protein interaction successfully. And I sort of just want to kind of show off some more stuff from eVOLVER. So like I mentioned before, we're trying to like make this thing easy for people to use. And so a big work that I did during my PhD was trying to make some graphical interfaces to sort of make it easier for people to pick up and use the tool. There's ways of interacting with the device. So on the top left, you have a way to just set up your experiment, interface with it directly, turn things on and off, see what's currently coming on and off the system, doing calibrations, viewing data, managing your experiments. And this is all stuff that's also changing. And like, I think they just got a grant to push this work forward a bit more, because I left and like I was the only one working on this. So now there's they have professional software developers now who are trying to make more UIs for the system, make it better. Other hardware developments that are happening. So this is something I worked on while I was there, just demonstrating that you can make new modules for this. So luminescence is something that's important for PACE, which is why I worked on this. But I made a little module that can detect luminescence within the device. I just made a new 3D printed part, made this little hardware system and slapped it on there, and I was able to get the data on and off of it. Pretty straightforward. I didn't have to like do any coding off of the Raspberry Pi. I just had to make an embedded, like my C code on the little microcontroller. That was all I had to do. And boom, it's sort of working. People have done this for other things too, like growing algae. There's a lab at Harvard that's doing that, using eVOLVER for algae growth. Yeah, like I said, people have integrated flow cytometers with it. People are doing even on vial fluorescence. There's a lab that's been working on that. Sort of similar to how the KaiBio has a system, which is KaiBio is another continuous culture or platform that's open source that you can purchase. It has it built in. People are making that also for eVOLVER. So the system is like sort of ready to go for like configuration and changing, which is the nice part of working with it. Lastly, I just wanted to highlight, there's a big community around this platform. So there's a forum where you can ask questions. There's plenty of guides and tutorials. There's a wiki as well that's constantly being worked on, and it's growing, trying to document more. How do you use the system? What can the system do? Tips and tricks, how things work. That's constantly getting better. And there's also a video guide for how to use it. So this is some of the work I showed earlier in the talk, but we did a Jove video paper about how to use the system and how you can use it to do a fitness parameter suite. And yeah, that's all I got. People have questions, so I'm happy to take questions. I know I sort of went very, very quick, but I wanted to leave time for people to have questions. So
nailed it. Let's all thank Zack before questions. A little visual round of applause. Thank you, Zack. That's awesome, and a ton of information. That's remarkable.
Yeah, there's a lot going on. Like I sort of trying to touch on a lot of stuff, but yeah, there's the cool thing I think is what people are doing with it now even more. Like I talk with some of my friends at the Khalil Lab, like the stuff for the anaerobic eVOLVER is really cool for studying gut microbiome. I think that's really, really exciting. And then some of the collaborations with other labs that they have going on are also pretty cool. So
we can go through the chat here. James, you did ask a question midway through about per each vial, how is external devices connected to the motherboard? Do you want to speak to that?
Yeah, so per vial, how is it connected? So there's basically a ribbon cable. So each smart sleeve has 16 pins on it that most of them it's like signal ground kind of thing. And there's so that just connects to a motherboard, and each one of those goes off for each parameter. There's an Arduino microcontroller. So let's say for stir, right? There's these computer fans, which is basically you're doing a PWM signal to modulate the computer fan speed. There's a Arduino that's controlling that for all 16 channels, which goes to a you actually have a slide. I can kind of go back to it. Do this. But okay, my computer froze now. Great. Come on.
I mean, the slides are still moving. Okay,
There we go. There we go. So you can see each so for the fan example, right? There's a slot on the motherboard. So the far left, there's that red Arduino. There's a multi-plexing PWM circuit there that can then send off your PWM signal for each vial through this PWM chip. And so temperature is the same way. There's a PWM signal. For signal coming in, there's an ADC circuit, which is basically just taking the signal from like a thermistor or whatever you have on a circuit to translate that voltage into an integer basically and send that to the Arduino, and this Arduino then takes that information per vial and it sends it off over serial to the Raspberry Pi, and then the Raspberry Pi has a network connection to your experiment code or any dashboards that you have that are reading that data off. Yeah, and we say stream to cloud here. It's just that it's connected to a router. at the end of the day. But yeah, hopefully that answers the question.
Cool. Yeah. I'll keep cranking through, but if someone wants to just, put up your little raised hand or something to ask it verbally, otherwise I'm just going to go down the chat.
Yeah, I can't see the chat for Zoom.
uh,
My computer.
Derek, you've preempted the line by being willing to speak verbally. So go ahead.
Yeah, great. Thanks. This is really cool stuff, Zach. I've been pouring through your thesis in the background as well,
Cool.
which is also kind of wonderful. So, in coordination with Church Lab, in this How to Grow Almost Anything course that I'm working on, we're doing a project on bacteriophage. And from an automation perspective, plaque assays are a real pain in the butt. So trying to figure out a system ideally to run in coordination with all these robots I was talking about at the start, the Opentrons robots, to enable a metabolic mechanism for monitoring the progression of bacteriophage. The two things that I really depend on for doing that are dissolved oxygen and glucose measurement, both of which are a little bit harder to do in this kind of shrunk environment than are things that you can access optically. Do you have any thoughts on how, particularly the micro-eVOLVER kind of system, which is what I'm most excited about from your talk here, might get shrunk down into something we can either run on platform on the OT2 or next to it in a way that would work in coordination with that?
So a couple things. One is I'll say we've actually hooked up an eVOLVER to an OT2 before.
Cool.
So we stuck it inside, and then we had an undergrad actually programmed it to be able to like interface with all the units for like sampling or doing things like that. So that's one thing, is that it is possible. People have done this before.
Nice.
You can, and I think OT2 is all Python as well, so you can definitely hook that up to the eVOLVER sort of script to include OT2 sort of interactions in your experimental algorithm if you wanted to do that. The other things I'll say is there is work currently to like do things in eVOLVER for like dissolved oxygen as an example. Like there's current work going on in the Khalil Lab to integrate that as a thing that you can do on a per vial, so on a 16 vial platform, being able to get DO for your vials, or in a bulk way too is another way that they're trying to do it. Depending on what you're trying to do, that might be useful. So that work's kind of ongoing. If you're interested in that, you could always reach out, or I can connect you to the guy who's working on it. He's a good friend of mine.
Well, that would be wonderful.
And then there was one other thing that you mentioned, but
glucose. And
glucose.
So my hack on glucose right now is to use test strip based. There's with all the continuous glucose monitors around, there's got to be a way of rigging CGM into a.
So actually, I'm glad I'm on this slide. So there's another thing you can do with the eVOLVER. So a cool thing, this was in the original paper, was millifluidics, which is a little bit more advanced. Like the way they make these, it's basically acrylic that's laser cut. And then there's a silicone membrane that you can then actuate using the eVOLVER to as like valves. So if you have a pump, you can then do things like route fluids through your valves through some sort of continuous measurement system if you'd like to, on a per valve basis. I don't have a good video here, but like I've done this for PACE. This is how I actually did a lot of my small molecule induction systems, is I used a millifluidic device. There's also been people playing around with the idea of like, doing a thing where I have a single measurement unit, like a flow box that I want to like take, let's say luminescence, just as an example. Like I have a luminometer. I can flow per vial into that device and then correlate that data back over like RS-232 or something to the system to then associate it with my experiment. So those are like the ways. There's lots of ways to do this. I think it kind of just depends on your needs and then how much work you're willing to put into it yourself. And I know a lot of these problems, like the Khalil Lab is still kind of actively thinking about. And if you're curious about using it or have ideas, like they're always looking for, cool ways to use eVOLVER and to collaborate. So I would encourage you to reach out to them, or I can connect you if you'd like to.
Yeah, fantastic, excellent. Thanks so much.
Yeah, no problem.
Cool. I see 2 more questions in the chat. I think we have time. Wait Sing asked, how much does it cost per day to run ePace?
Oh, that's a good question. So that comes down basically to media costs for ePace, right? So there's a triple antibiotic selection for the experiments that we were doing. So antibiotics was the predominant cost of what we were doing. So I was making media to run 2 platforms simultaneously, I was making about 40 liters of media every couple days. And that's a couple grams of each antibiotic into each of those carboys, basically. So I think that's the main cost. I did run the numbers at one point. It can get kind of pricey with the antibiotics to run ePace at that scale. But it's so yeah, if you do the numbers with the antibiotics, it's probably a few $1,000s for like a week of running. The system itself doesn't cost anything really to run. It's just the electricity to keep it on, but it's not like consuming massive amounts of power. So it's really just your media as the main cost driver there.
Cool. Still quite good for, you know, if you get a new strain out of it.
Yeah.
Final question. Garrett, you asked about bioreactor. Do you want to add any more context to that?
Yeah, well, I was just wondering if Zack was familiar with bioreactor. They're a newer open source project that it really looks as if their design may have been may have taken some inspirations from your project.
Yeah, actually, I think it's awesome that like there's all these other platforms that are popping up now. That like I think they're borrowing and also making some things better. There are some things I think eVOLVER does better than other. Like I think the scalability and the way you can interact with large numbers of vessels at once, eVOLVER does a really good job of that. I've used the Kai-Bio system recently, and I found it much more like it's still great. Like I think a lot of the hardware on it is actually higher quality than eVOLVER, so like you can get fluorescence out of the box, and like you get different types of temperature, and it's a very nice system for what it is, but I find like the scaling of it was actually harder. So I was just running 8 vessels with that. I found that a little more challenging because of the way that they have the way the platform is sort of set up. So I think, you know, all these different open source platforms and things that people are making, I think they all have a place, and depending on what your lab is doing or what you're trying to do, you know, one might be better than the other. I think they can also all learn from each other. I actually think, like, you know, I've reached out to some of the Kai-Bio people on their forums asking questions, you know. So you know, the more that these things sort of pop up and the more people work on it and like learn from each other, the better, in my opinion. So I'm like super excited to see things like the bioreactor and, you know, Kai-Bio, and I've seen some other papers popping up of like one-off systems that people are making that take inspiration from all these systems for their, you know, to solve their specific problem too. So you don't necessarily have to like, I need to go get an eVOLVER. You can, you know, just all this stuff is open source. The hardware is all open source. You know, everything's sort of open. So people like learning from it, making your own to solve your own problems is also, you know, super cool.
Awesome. Well, we're at the top of the hour. Isaac, I see Isaac has a question about silo compatibility, but maybe we can take that offline.
Oh yeah, we've played with that. I think that was right before I joined the lab. They played with that, but yeah.
Cool. All right. Well, let's thank Zack one more time. Zack, this is so cool. It's such a great piece of work. So thank you for presenting it. And
thanks for having me, Tim. Also, yeah, it was great to talk about eVOLVER again. Yeah.
yeah. We hope you'll come back. Thanks, guys. Cool. So I'm going to put this video. In fact, I'm going to stop the recording.